Yes, that is a very good starting point for our, interview. When ISEF, the International Society of Extracellular Vesicles, was created back in 2012, and we held the first international meeting in Gothenburg in Sweden, I was able to meet several Spanish people and scientists working on extracellular vesicles in different fields. So I thought of the idea that perhaps we should create also our own society to do research on extracellular vesicles. So when we went back to Spain, we first made a meeting in Barcelona. And a few months later, we were able to create the first Spanish group of investigation and innovation on extracellular vesicles, whose name is now JVEX. Now, since the moment that we created JVEX, our philosophy was and still is, and I hope that it will continue to be, that any money that was earned by the society from congresses, from workshops, from the courses, etc., will be devoted to mobility fellowships that will allow fostering scientific communication among the students, postdocs, etc. And this has been done with great success. We are now on the 10th call of these mobility fellowships, and we have more than 27 students and postdocs that have profited of them. Now, after the JVEX Society was created in Spain, the movement, this idea of getting other European societies started to grow up. And after that, the Germans decided to create their own society, the GSEF Society, whose president was at that time Dr. Bernd Giebel. And it was followed by the United Kingdom, who created the United Kingdom Extracellular Vesicle Society, UKEF. At that time, also, the president was Dave Carter. And I have a long-standing collaboration and friendship with Bernd Giebel. And together with Dave Carter, we met in JVEX. I think it was the 5th JVEX meeting that we did in Granada back in 2019. And during the evenings, having some beers, we discussed the idea that perhaps we should, elevate this idea of the mobility fellowship to the 3 societies and start, fostering collaborations among the people in our different societies. Now, the idea really took off. And after that, I retired from the president of JVEX. So Dr. Maria Yanez took the presidency. And the same happened with the United Kingdom Society. Dr. David Carter retired, and it was replaced by Dr. Charlotte Lawson. Bernd continued at that time to be the president of the German Society, and we joined with a new society that was created. It was the Italian Society, EVITA, which was the president is Dra. Benedetta Bussolati. And these 4 people really decided to take the lead to really make this MOVE meeting in Malaga a reality. Also, they counted with the enormous support of Dr. Maria Isabel Lucena in Malaga and all the team, as well as the university, which facilitated all the venue. As the idea of MOVE was growing, we were very inclusive in communicating this to new European societies of extracellular vesicles that were being created. And at the end, amazingly enough, we came out with 15 different extracellular vesicle societies in Europe that were all willing to participate in Malaga. And there is a nice anecdote about the name, MOVE, which means Mobility for Vesicle Research in Europe. It was suggested by Dr. Benedetta Bussolati, and it was unanimously accepted. So I think this is the history of this MOVE meeting, with the idea of foster mobility fellowships among the different people working in this field in Europe. And I am going to facilitate to you, which is freely downloadable, an animation that was made in order to illustrate the creation of the societies. And you're more than welcome to put it together with the interview for anybody who's interested in downloading the story, okay? It's an animation. That's a difficult question, I don't like to really say, but I have to say that I was really surprised. I mean, the meeting really surpassed all my expectations. 400 people, so many sponsors, so many good environment, etc. And I didn't know nothing about the price. Honestly, this is the honest truth. So when I was sitting there in the final ceremony, when Bernd Gebel and I was called to the stage, it was a tremendous surprise. And I was feeling very honored and very happy, you know, because it is true that I ignited and catalyzed this idea together with Bern and Dave. So having this recognition from you has been very special. I hope that you keep the EVCNA move prize, because it's a very incentive prize, you know, for the meetings. And yes, I really thank you, because it's one of the best prizes in my career. Honestly, it's symbolically, in the heart. It's for me. It was a very important prize. So thank you so much. Thank you for this question. You know, working with malaria is really a scientific passion of my life. I've been always working with malaria, but unlike what most people think, I'm not working with the malaria that is mostly present in sub-Saharan Africa, which is caused by Plasmodium falciparum. I'm working with a neglected human malaria parasite that is called Plasmodium vivax, and it is highly prevalent in Latin America, the tip of Africa, Madagascar, and East and Southeast Asia. It is believed that around 2.3 billion people are at risk of this disease, and last year it was calculated that around 4 to 5 million clinical cases were reported. But this is certainly an underestimate, because we now know that more than 90% of the infections are chronic and asymptomatic. And for years, it was thought that these chronic asymptomatic infections were due to dormant stage of the parasite in the liver that is called the hypnozoite. But I think everything started to change back in 2001. Can you imagine how many years have passed? When I was at that time an associate professor at the University of Sao Paulo in Brazil, and together with Dr. Camer Fernandez Becerra, we discovered that this parasite contained variant surface proteins at the surface of the infected reticulocyte. And when we discovered this multigene family, because we know a lot about multigene families in Plasmodium falciparum, we postulated that perhaps in the case of Plasmodium vivax, there was also cytoadherence, which could be responsible for pathology. At that time, there was no belief that there was cytoadherence in Plasmodium vivax malaria, but there were several groups, including ours, that they started to show some solid data that cytoadherence was feasible in Plasmodium vivax. But at that time, I think what was very important was the animal models. There was an animal model of Plasmodium vivax. It was a rodent malaria parasite that's similar to Plasmodium vivax, invades reticulocytes, which are very young red blood cells. And looking at this rodent model, Professor Leon Weiss in the United States showed some images that really impacted us. The images showed that the infected reticulocytes in this rodent malaria model cytoadhere to the spleen. And this was a little like a heresy, because the spleen has been always considered the filtration organ where malaria parasites are destroyed. But in the case of vivax, we postulated at that time that the cytoadherence could be inside the spleen in order to create cryptic intrasplenic infections. And this was very difficult. I have to tell you that trying to demonstrate this was very difficult. There was an enormous scientific untrustness. Nobody believed that could be feasible. The spleen was there to destroy the parasites, and to have a parasite that hide inside this organ was not feasible. But we demonstrated proof of principle with a rodent model. But again, when you work with rodent models, there is a lot of skepticism with what is happening with the human condition. And so in 2012, we have a serendipitous case of a 19-year-old boy who fell out of a tree. He was perfectly under health conditions. And at the time that he fell out of the tree, he suffered a spleen trauma. The spleen ruptured, and it had to undergo surgery. So the spleen had to be removed. And so for the first time in the clinical literature, we were able to look at a spleen that had not been treated with any antimalarials, and try to look if there was any evidence that the spleen contained parasites. And to our surprise, it was fantastic. We published this data in close neglected diseases. We were able to, for the first time, demonstrate that the spleen was absolutely packed with intact infected reticulocytes. And then to answer your question about extracellular vesicles, I started to read in Brazil all this fascinating field of extracellular vesicles, starting with the seminal publications of Rose Johnstone's and Philip Stahl, describing that reticulocytes mature into full erythrocytes by releasing extracellular vesicles that they call exosomes. And because of the fact that reticulocytes are the host cell of Plasmodium vivax, we started to postulate that maybe the extracellular vesicles coming from natural infections could contain some signals that will favor all of these intrasplenic infections. As you know, the field of extracellular vesicle has exploded. There has been many publications. Grasa Raposo demonstrated that they could be used, in as vaccines. The work of Jan Loveball showing that they were involved in intercellular communication, etc. So I wrote our huge proposal, proposing that the EVs from natural infections were involved in the intrasplenic cryptic infection of Plasmodium vivax. Unfortunately, the grant was not granted. And at that time, that was 2015 or so, Professor Pedro Alonso here in Spain had a discussion with me and offered the possibility of coming to work in Spain in a new research institute that at that time was called CRECIP and now is part of the Institute for Global Health, is global. And so I came to Spain. I had the enormous luck of participating on a public context, and I won a career research position. And ever since, I've been working in trying to demonstrate that extracellular vesicles from natural infections indeed are able to cause these intrasplenic infections. We have had very nice publications, once in Nature Communications that shows this very elegantly. And most important is that we collaborated with Professor Nick Anstein in Australia, and we were able to publish in New England Journal of Medicine and PLOS Medicines that indeed the spleen of peoples living in endemic regions of malaria contain cryptic infections during chronic infections. And again, I will share with you a slide that points out to this new cycle of Plasmodium vivax. And I think this really covers your question in great depth, and thank you for the question. Yes, that is a long and winding road story. But back in 2014, I co-ascribed to the German Trias and Pujol Research Institute, IGTP, where I physically moved, and I'm still working here. One of the reasons that I decided to physically move here was because the director, the former director, Dr. Manuel Puig, had this vision of innovative research that could really make a translation and impact. And this institute really enforces the fact that you can create spin-offs out of your ideas, and they really support you about that. So together with Dr. Francesc Borràs, who's a researcher here at IGTP that is working with extracellular vesicles and kidney failure, we decided to create this spin-off that we called Innovex Therapeutics, SL, and it was born in 2014. And for the past almost 10 years, since the creation of this spin-off, we have been trying to raise capital risk, risk capital money in order to develop the vaccine against Plasmodium vivax. And we have been able to, as I said, demonstrate the proof of principle in a rodent malaria model that extracellular vesicles from this rodent malaria model, when you use it as a vaccine, it really protects the mice from infections. But then really to raise the money for the human trials is not easy. We have not been able to really convince, most of the agencies that it will be worth to go into, phase 1, 2, and 3 trials of Plasmodium vivax because of the fact that there is already a vaccine against Plasmodium, no one but 2 vaccines against Plasmodium falciparum, But still, I think it's a mistake because the vaccines are species specific, so that the vaccine that is working against Plasmodium falciparum is not working against Plasmodium vivax. And if we want to eradicate malaria, we're going to really have to pay attention. So we continue on the search for this. At the same time, we also target another disease because a company needs to have different products. And the product was to develop a vaccine based on extracellular vesicles against a virus. But this was a virus that caused disease in animals, specifically in pigs. It's called the Pierce virus. It's the pig reproductive and respiratory virus. And the story here is very nice. We have been able to create a platform for developing and creating and producing extracellular vesicles with viral peptides. We have gone into the proof of concept that we have, farm clinical trials that have protected the pigs. And we have now sub-licensed this product to 2 major Spanish companies that will make it in a large scale and will make this result. So I think it's okay. I think I have to warn the young people that will listen to this, that it's not an easy road, but I think we have to really think more and more and more in spin-offs, because it's another solution to the bottleneck that we have with academia. And at the end, it's just, serendipity. And if your idea flourishes, it's okay. If it doesn't flourish, you have to accept it and you move to another one. And so the company is still there. We don't know if the vaccine against pigs will, after it's being raised in large-scale production, will protect, but we certainly hope so. And we continue trying to look for funds to develop the vaccine against the Plasmodium vivax malaria. So I think this is more or less the story of Innovex, okay? That's not an easy question. I guess the first thing that I would like to do is to reinforce to them that they're entering an excellent field. Extracellular vesicles research field is intellectually rewarding. You can have lots of molecular insights into what happens into normal and physiopathological conditions. It has an enormous translational potential in terms of therapeutics, in terms of drug delivery, in terms of biomarkers of disease. So I think they're entering really a fascinating field. However, I have to say that the field is complex and it's not easy. And the complexity is because extracellular vesicles have an enormous wide variety of, of sizes, properties, composition, and that is all in the same biological fluids. It doesn't matter if you use tears, if you use urine, if you use plasma. When you get extracellular vesicles from this milieu of things, you have really a heterogeneity that is an unbelievable complex. We don't have a particular marker that is specific of each particular cell that we can try to use immunocapture to really get, the most out of, particular cell types that we want. We're progressing a lot into different technologies, but still we have an enormous confounding factors with the extreme complexity that they have in circulation. Obviously, we are working also with in vitro culture, but I would like to share with you the fact that in proteomics analysis that we have done with in vitro culture versus extracellular vesicles obtained directly from the plasma of patients, there is very little intersections, saying that perhaps the in vitro culture proteomics and molecular cargo characterizations and functional analysis might not be reflected in what is happening really in the real infection. And so in that sense, I really think that people entering this field have to be extremely critical in running specific controls. When you do, for instance, uptake experiments, you have to do those concentrations, you have to do cell specificity, you have to demonstrate this in the milieu of the complex organs, etc. The challenge is huge, but I think it's important for them to know that it's a fascinating field. We have also another recent example. We did the proteomics characterization of extracellular vesicles from SARS-CoV-2 patients at the beginning of the pandemic, of COVID pandemics. And there are so many so many contradictory and contrasting results in the literature about the proteomics of, samples from COVID patients that exemplifies the difficulties that we have. So happily, myself, is now, will publish soon again another guideline, etc., that really makes rigor and standardization on the extracellular vesicles isolation, purification, characterization. But the important thing for the newcomers is critical mind, to be really critical, to question everything, to run the controls, to know exactly what are your samples, and especially it would be important to reproduce. I think one of the main problems in the fields right now is that when you get a publication and you try to reproduce these results in your lab, many times you fail. And so before the publication, it would be important to have the rigor, the scientific rigor of reproducing themselves, the results, to be sure that it is a robust result that can be shared with the community. But again, I would like to finish by saying that this is a fascinating field. We have global warming, vector-borne diseases are going to change. Studies of extracellular vesicles in vectors is in its infancy. The origin of extracellular vesicles together with the origins of viruses. I mean, there is a huge field that is ahead, and I think is fascinating, and I would be one of the main pushers of students, new students to come into this field. Okay? Well, obviously, because of the fact that we were able to postulate these spleen infections, and also more recently, we have demonstrated also that the parasites have cryptic infections in the bone marrow. This was not our original idea, but there were a lot of conflicting results in Plasmodium vivax, and we were really able to demonstrate unequivocally that the parasite is also present in the bone marrow. So we will really like to unveil the molecular basis of these cryptic infections and the role of extracellular vesicles in making these infections. It's very interesting because we have preliminary data showing that extracellular vesicles from infections really send different signals to the bone marrow and the spleen, and I believe that we will be able to contribute to unveil this. So this is a very basic research question that I would really like to pursue. Now, one of the main problems that we have with this parasite is that we cannot culture it in vitro. Unlike Plasmodium falciparum, the parasite responsible for malaria, human malaria in sub-Saharan Africa, most in sub-Saharan Africa, there is no in vitro culture of Plasmodium vivax. And there has been more than 120 years of many different labs trying to develop the in vitro culture. We don't understand why we have tried, and people have tried many things, but we have failed. So in order to advance into this, and especially in working with the bone marrow and the spleen, which ethically and technically is very difficult. If you have an infection, you cannot touch the spleen because you will cause a fetal, fatal hemorrhage. Or the bone marrow, we all know that is a very aggressive, invasive technology. So we have resorted to two different solutions. One is we are trying to develop organs on a chip, which was what I presented in Malaga. We are trying to really use organs on a chip to emulate minimal functional units of the bone marrow and the spleen, and this will facilitate the studies of the role of extracellular vesicles at a speed and a velocity that will allow us to look at what happens in those cells. Obviously, this is a very reductionistic approach that will have some limitations, and therefore we also are trying to develop a humanized mouse model of the human bone marrow and the spleen. And we have gotten the ethical permission. These are not easy experiments to carry on, but we have finally gotten the ethical permission, and we are now starting to try to develop these humanized mouse models of the human bone marrow and the human spleen, which will facilitate really a much more in-depth analysis of the role of extracellular vesicles in these infections. On the translational aspect, obviously, pursuing efforts in terms of the vaccine against Plasmodium vivax. In fact, with Bernd Giebel, my good friend, we have a standing collaboration on this. We have tried to get some grants, and we are now collaborating together in several ideas on how to target, the Plasmodium vivax proteins into extracellular vesicles, etc. So we will continue on that. And on another aspect that we have also started some years ago, this was in collaboration with Stefan Kappe at Seattle's Children's Hospital in the United States. We are trying to discover biomarkers in circulating extracellular vesicles of patients with cryptic infections, because if we are not able to identify those patients carrying chronically and asymptomatically the parasite, the malaria transmission will continue. So I think this is it. I mean, I think the lab has a unique basic biological questions and translational questions, and this is what I would like to pursue for the next 5 years.